Hello, good day to everyone. This is Dr. Yadnik from KEM Hospital, Pune, and I run the diabetes services there. What am I doing as a diabetologist in a meeting of micronutrients? That's because our research concentrates on developmental origins of health and disease, especially in relation to diabetes, and the role micronutrients play in this important new medical concept. Our logo is a mother and a baby because we believe that this unit determines the fate of the child for the rest of the life. And this modern Ganesh, who is lying down comfortably on couch, ignoring the mouse who had served him all his life, but now replacing that by clicking the mice of the computer. A rapid transition over the years. Simply said, DOHAD is developmental origins of health and disease, and this is a new concept in medicine. While I was training as a medical student and as a postgraduate, we were taught that non-communicable disease have a genetic predisposition and lifestyle precipitates it. This conundrum has now been challenged by the DOHAD concept. How did it start? Though there were indications before that early life was important for future health and disease risk, it was David Barker who published this remarkable paper in 1991 and focused the attention. He showed that lower the birth weight, higher was the risk of diabetes and metabolic syndrome in adults. He called it fetal origins of adult disease. And he came and met me in 1991 and explained this to me, and I was thrilled. I'm proud to say I suggested the name thrifty phenotype for his hypothesis. And what was that? That was the hypothesis that type 2 diabetes is an outcome of the fetus and the early infant having to be nutritionally thrifty because it didn't have enough nutritional supply in utero. Work in humans and especially work in animals showed that such an association between poor early life growth and subsequent risk of diabetes and other non-communicable diseases was due to epigenetic reprogramming. And this was a very exciting idea, which immediately brought in experts from different branches into this new and exciting field. Very soon, it became apparent that epigenetic programming, though largely very active in utero when development and differentiation occurs, does continue into postnatal life. And there are additional windows in the postnatal life where epigenetic reprogramming can again occur. And this was then incorporated in the idea of developmental origins of health and disease. And I call it an ongoing iterative program written across the life course. Starts from before and the conception, continues through pregnancy, childhood, puberty, and adult life. In a way, it's never too early, never too late to intervene. And that's the nicest thing about it. Soon, this was now taken in the field of development and differentiation, and the concepts in that field, like plasticity, programming, and teratogenesis, were applied. If a developing system has plentiful surroundings, it develops to the full freedom of its growth and development. If there is a restriction in the environment, deficiencies of some sort, there is a restriction of growth, restriction of plasticity, which is what we call programming. It leads to structural and functional imprints which remain with you for the life. But remember, the windows of vulnerability and therefore of opportunity are maximum in utero and in the other windows of puberty and childhood growth, which we have seen before. Both genetic but especially epigenetic mechanisms operate, and the stimuli could be nutritional, maternal metabolism, maternal stress, and a whole lot of pollutants and other unknown substances which might reach the fetus through the maternal circulation. So, one aspect of nutrition is, of course, micronutrients. And micronutrients are important, especially in pregnancy, because that is where they play an active role in the development and differentiation. The best known example of role of micronutrients in the development and differentiation of fetus, which might have disastrous effects, is the famous neural tube defect story. This was result of lot of basic work in animals and humans and led to two seminal trials which showed the importance of folate and B12 and other micronutrients in the story. In the MRC vitamin study, recurrence of NTD was reduced by almost 80% if the mothers were supplemented with 4 mg folic acid starting before pregnancy and continued in the first trimester. The first occurrence NTD was completely prevented in the Hungarian trial by a vitamin mixture of 12 vitamins and minerals. It contained 4 micrograms of vitamin B12 and 800 micrograms of folic acid in addition to other contents. The control group contained some minerals only. And this was a dramatic trial which showed complete prevention of neural tube defects and reduction in other congenital abnormalities in the Hungarian women. India is one of the countries with largest incidence of neural tube defects, anything between 3 to 11 per thousand deliveries. And we have some experience in this. We did one of the largest studies of neural tube defects in the world in 4 centers in India. We studied over 300 neural tube defects and compared them with almost 700 non neural tube defect pregnancies. What did we find? The concentrations of vitamin B12 and folate in the mothers were no different whether they had given birth to either a neural tube defect or a non-neural tube defect baby. On the other hand, holo-TC, the active form of vitamin B12, was lower in the mothers of babies who had neural tube defect, and this also reflected in higher homocysteine concentrations in these mothers. To further clarify the role of B12 against folate, we did genetic studies in the mothers and showed that MTHFR, a gene which regulates folate supply for the methylation of homocysteine, was in fact actually protective in the Indian mothers against the neural tube defects. On the other hand, a mutation or an SNP in TCN, transcobalamin 2 gene in the mother and also in the baby, predispose the baby to neural tube defects, suggesting that in the vegetarian Indians, the neural tube defects might also be driven by B12 over and above the well known effects of folate. After that dramatic example of a neural tube defect predisposition by micronutrient deficiencies, I'm going to discuss now role of micronutrients in fetal growth and future risk of non-communicable disease in a Dohan model. This slide shows you two interesting cartograms. That's the world maps where size of the country is proportional to the burden of the condition which is being described. India, as you know, is one of the capitals of diabetes, and traditionally we consider diabetes as a disorder of overnutrition. At the same time, India is an even bigger capital of undernutrition in early life, measured either as low birth weight or under 5 undernutrition. And David Barker's work allowed us to think that these two could be related to the undernutrition of the fetus because the mothers would be undernourished and micronutrient deficiencies are part of the maternal undernutrition in India. We started our Pune maternal nutrition study in 6 villages near Pune in 1993 and studied 800 or so pregnancies in the mothers whom we had followed from before conception. We measured the babies in detail in utero with ultrasound. We studied maternal nutrition and metabolism. We measured the babies at birth, then every 6 months till 18 years, and every 6 months we have done their cardiovascular and body size and body composition measurements. Our 24-year follow up was halted by the COVID epidemic in February. We have an impressive rate of more than 95% follow up in these studies, which is quite remarkable. And we have a biobank of samples on the mother, children, and the families going back to 1993. The purpose of the Pune maternal nutrition study is to study the nutritional regulation of fetal growth and follow up the offspring through their life course to see the evolution of non communicable disease. The average mother in PMNS was 42 kg with a body mass index of 18.1 at the beginning of pregnancy. She was active in the field and at home and consumed less than the recommended dietary allowance for macronutrients and micronutrients. She produced babies which were on average 2.7 kg, and up to 70% of these babies could be classified as small for gestational age. We did not find an association between maternal macronutrient intake and fetal growth, but a strong association between foods which are rich in micronutrients like green leafy vegetables, milk, and fruit. We also found a strong association between maternal circulating folate concentrations and birth weight of the baby. And interestingly, higher the maternal homocysteine concentration, chances of baby being small for gestational age were very high. Where did this high homocysteine come from? Folate deficiency was very rare, but B12 deficiency was present in two out of three mothers. Interestingly, these babies, which looked small, thin, and scrawny, when we compared them with the English babies, we learned quite dramatic things. The Pune baby was 2.7 kg, English baby was 3.5 kg, so 800 grams heavier. But the Pune baby had extra fat compared to the English babies, so that in the subcutaneous compartment and all the three compartments of the abdomen shown here on an MRI scan were higher in the Indian baby. And this allowed us to develop the concept of a thin fat Indian baby which had higher concentrations of leptin and insulin in the cord blood and lower concentrations of adiponectin, all these factors suggesting increased risk of diabetes in later life. We followed up these babies after their birth. We made some very interesting observations. Maternal concentrations of B12 and folate in pregnancy predicted the adiposity, the insulin resistance, and the neurocognitive performance of the child right up to young adult age, that's 18 years. Mothers who had low B12 but high folate in their blood during pregnancy produced babies which were more adipose and insulin resistant. Another vitamin which contributed to this association was low vitamin D in the mother, which increased the adiposity of the baby. And low B12 in the baby affected its neurocognitive performance in different domains. And now, in an exciting finding, we can see certain areas of brain underdeveloped in the children of the B12 deficient mothers. A profound effect of intrauterine undernutrition on subsequent health. Our results told us to concentrate on one carbon metabolism, and so we learned more about it. This is transfer of a methyl group across different reactions. And there are more than 80 methylases in the cell, so it must be very important. And we soon realized that this metabolism affects DNA synthesis, repair, its stability, and very interestingly, methylation of DNA, which is an epigenetic regulator of the gene expression. And we have demonstrated in B12 intervention studies that methylation of genes related to either adiposity or diabetes can be demonstrated in human beings. A very important concept which I think is going to influence the thinking in this area. We soon realized that one carbon metabolism is very important in the periconceptional period as shown in this cartoon because so many processes happen at that time like gametogenesis, the fertilization, its implantation, embryogenesis and placentation. These are profoundly affected by the one carbon metabolism and therefore preconceptional and periconceptional one carbon metabolism becomes an important issue if we want to. What about other micronutrients? Though we may not have direct proof of this in Pune maternal nutrition study, there is vast literature to highlight the role of vitamin A, iodine, vitamin C, vitamin D, fatty acids, iron, B12 folate, and the boxes where I have shown a red color, like vitamin A, folate, B12, vitamin D, omega fatty acids. These micronutrients are genoactive in some way. They will influence either gene expression or some other function of the genes. They become very important for the health of the individual, not only the adults, but the baby growing in the mother's womb. I'll tell you another interesting thing. We think of mother and baby as a dyad, but if a mother is pregnant with a female child, I don't know how many of you know that the female child has all the 1 million ova it is supposed to have by 20 weeks of its gestation. We have this unique situation that in a pregnancy with a female child, you have the grandmother, the daughter, and the grandchildren all exposed to the mother of the grandmother. A very unique situation which will influence the health and disease of three generations and a unique opportunity to improve the life of three individuals. Based on our findings, we have proposed a nutrient mediated teratogenesis idea for B12 and folate, suggesting that structural and functional changes in the developing fetus, which may manifest in growth restriction, limited reserves, and subsequent risk of non communicable disease. So that is a very interesting. Finally, I am going to advertise a film we have made about mother's nutrition and its long-term effects on the baby. and that's on the YouTube channel of the KEM